In this question, you have to identify the wrong statement regarding restriction enzymes. Now, restriction enzymes, also known as restriction endonucleases, are found in bacteria and archaea that recognize and cut DNA at specific sequences known as the recognition sites. And these enzymes break the phosphodiester bonds in the DNA backbone, creating double strand breaks. And these enzymes are crucial in genetic engineering and recombinant DNA technology, allowing precise DNA manipulation by cutting at specific sites. And they produce sticky ends or blunt ends and can be used for cloning or gene modification. Now, coming to the options. Option A, they are useful in genetic engineering. As already mentioned, this is correct. Hence, this is incorrect option. Option B, sticky ends can be joined by using DNA ligases. This is also true. Now, DNA ligases are enzymes that can join the sticky ends of DNA fragments after they have been cut by restriction enzymes. Hence, this is incorrect option. Option C, each restriction enzyme functions by inspecting the length of a DNA sequence. Now, this is wrong. Restriction enzymes do not inspect the length of a DNA sequence. Instead, they recognize short sequences of nucleotides, usually 4 to 8 base pairs long, known as recognition sites, which are often palindromic, and they cut the DNA at these sites. Hence, this is the correct option. Option D, they cut the strand of DNA at palindromic sites. Now, this is true. Restriction enzymes usually recognize and cut the DNA at palindromic sequences. That is, the sequences that read the same forward and backward. Hence, this is incorrect option.